Good day, everyone. Today we're diving into experiment number 10, hydrolysis of carbohydrates. This laboratory activity focuses on understanding how complex carbohydrates, specifically starch and glycogen, are broken down through hydrolysis, and how we can detect the progress of that breakdown using the iodine test. So, this topic is fundamental or it's foundational because carbohydrate metabolism is central to clinical chemistry and human physiology. By the end of this experiment, you should be able to first hydrolyze starch and glycogen, which are the two major polysaccharides. Second is to monitor the progress of hydrolysis using the iodine test. And lastly is to correlate your findings to how carbohydrate catabolism occurs in the human body. As a medical laboratory science student, understanding these reactions is essential for interpreting biochemical processes related to digestion and metabolism. Starch is the primary dietary carbohydrate for humans. It makes up over 50% of our carbohydrate intake. It exists in plants in the form of granules, particularly in seeds and tubers. So starch is a mixture of two polymers, amylose and amylopectin. Amylose consists of about 10 to 30%, and amylose is a linear polymer made of D-glucose units connected by a alpha 1,4 glycosidic bonds. However, it is not a straight chain. It forms a coiled helical structure with about 6 glucose units per turn. Inside this helix is enough space to trap iodine molecules, forming a complex that yields the characteristic blue-black color in the iodine test. This reaction is highly sensitive, detecting even minute amounts of starch. While amylopectin, on the other hand, consists of about 70 to 90%. This composition gives starch its structure, digestibility, and reaction with iodine. So there are experimental evidences which indicate that amylose is not a straight chain of your glucose units, but instead it is coiled like a spring, consisting of 6 glucose monomers per turn. And the characteristic blue-black color that appears when starch is treated with iodine, it is due to the formation of your amylose iodine complex. Amylopectin is a branch polymer. Most bonds are alpha 1,4, but approximately every 25 to 30 units there is a branch at an alpha 1,6. Although it also forms a helical structure, the branching disrupts the uniformity of the helix. So when iodine interacts with amylopectin, it produces a reddish-brown color, less intense than amylose. This difference in color helps us distinguish the composition of different carbohydrates. Glycogen is the storage carbohydrate of animals, including humans. You'll find it in almost all cells, but especially in the liver, 4 to 8% by weight of our tissue, and then skeletal muscles, 0.5 to 1%. So it appears as granules inside our cells, similar to starch granules in plants. Glycogen is the body's immediate reserve or glucose, especially during fasting or between meals. Structurally, glycogen is very similar to amylopectin, but it is more highly branched. Branches occur every 8 to 12 glucose units, as you can see in the picture. Because of this, glycogen reacts with iodine to form a reddish-brown color, not blue-black. Glycogen can be hydrolyzed by first is acid hydrolysis, or second via the enzymes such as your phosphorylase, which releases glucose 1-phosphate units. Salivary amylase is also called as your thiolin. It is the enzyme in your saliva that begins carbohydrate digestion. It works best at pH 6.7 to 7.0. It breaks the alpha-1,4 glycosidic bonds in the starch, and it produces oligosaccharides, maltose, or sometimes glucose, depending on exposure time. So this enzyme doesn't act on branch points, meaning glycogen and amylopectin are only partially hydrolyzed. So both starch and glycogen undergo hydrolysis, a reaction where water breaks chemical bonds. Amylase cleaves the alpha-1,4 bonds, producing shorter polysaccharides such as your dextrins, maltose, and glucose. If the reaction goes to completion, the final product is glucose, which then enters pathways like glycolysis. Although salivary amylase can act on glycogen, it is not very effective because glycogen is highly branched. The primary enzyme for glycogen breakdown in the body is glycogen phosphorylase, which works inside cells to release glucose phosphate units. With enough time, salivary amylase may show minimal hydrolysis of glycogen, but not nearly as effectively as with starch. So in this experiment, the iodine test is our primary indicator for hydrolysis. So how do we interpret this one? If there is the presence of blue-black coloration or a blue-black colored compound, then starch or amylose is present. Then if there is the appearance of reddish-brown colored product, then glycogen is present. And if there is a yellow-colored compound, then there are no polysaccharides, which means there is a complete hydrolysis happened during the reaction. So as hydrolysis proceeds, the color shifts from blue or brown to yellow, showing the breakdown of complex carbohydrates. So for starch, salivary amylase randomly hydrolyzes internal alpha-1,4 linkages, except the ones at the ends on those beside branch points. Starch degradation occurs in two stages. First stage is the production of branched oligosaccharides, and then the second stage is the formation of dextrins, maltose, and glucose. So remember, enzyme specificity means amylase acts only on specific bonds and only on substrates that fit its active site. For glycogen, amylase is not well suited to hydrolyzing glycogen because of its frequent branching at alpha-1,6 linkages, which the enzyme cannot attack. Even though glycogen resembles amylopectin, its branching pattern makes it physically inaccessible to amylase. Thus, hydrolysis is limited, and the iodine color tends to be reddish-brown, fading only slowly over time. So this part right here outlines the experimental method. You are going to perform a spot test preparing 10 samples each for starch and glycogen. To each sample, you add saliva, which is the source of the amylase enzyme, and then Lugol's iodine reagent at 5-minute intervals. So this allows you to track hydrolysis over time and correlate the color changes to the degree of breakdown. So this summarizes the expected results. Starch digestion progresses from blue, black to brown to yellow. Glycogen digestion shows a reddish brown color that changes minimally with time. Remember that Lugol's iodine is light sensitive, so it must be stored in dark containers. These are the recorded experimental results for starch hydrolysis. As expected, starch samples likely turned from blue-black to lighter shades and eventually yellow as amylase broke down the alpha-1,4 linkages. Comparing your actual data to theoretical expectations helps validate the reaction kinetics. And with that, everyone, I would like to conclude my post-lab discussion with experiment number 10, all about carbohydrate hydrolysis. Hopefully you have learned something from this video. So thank you so much for listening and have a good day.